The overall goal of this procedure is to demonstrate how to slice and dissect the suprachiasmatic nucleus from transgenic mice and to culture the suprachiasmatic tissue in order to record clock gene or protein expression as luciferase-induced bioluminescence. This is accomplished by first dissecting out the brain from a transgenic mouse that contains a luciferase reporter for a clock gene or protein, and then making a brain slice containing the suprachiasmatic nucleus. The second step of the procedure is to dissect out the small bilateral suprachiasmatic nuclei from the slice. The third step of the procedure is to culture the suprachiasmatic nucleus explant on a membrane in a petri dish that contains culture medium and luciferin. The final step of the procedure is to record luciferase activity by measuring bioluminescence with photomultiplier tubes in a light, tight heated chamber. Ultimately, results can be obtained that show circadian oscillation of the clock gene or protein.
My name is Sergey Savelyev and I'm working in Gabriela Lundqvist lab.
My name is Ann-Sofie Johansson and I'm working in Gabriela's team.
My name is Gabriella Schmitz-Lundqvist. I am a principal investigator at the Department of Neuroscience.
And my name is Karin Larsson and I'm a PhD student at the Swedish Medical Nanoscience Center and I collaborate with Gabriella.
The main advantage of this technique over existing methods like Western blot, RT-PCR and in situ hybridization is that expression of a clock gene or protein can be followed continuously for many days in the same tissue with very high resolution of about 1 minute, allowing detailed analysis of period, amplitude and phase of the circadian rhythm.
Visual demonstration of this method is critical, as the SCN slicing and dissection steps are difficult to learn. The SCN is very small and can be difficult to identify.
Demonstrating the procedure will be Dr. Sergey Savelyev, a postdoctoral fellow in my research team.
After making the necessary solutions, prepare the petri dishes that will be used in the experiment. Using a 5 ml syringe pre-filled with autoclaved vacuum grease, apply the grease on the top ring surface of a 35 mm petri dish. Prepare a separate petri dish for each slice culture. Fill a falcon tube with 10 ml of air-buffered culture medium. This will make enough medium for 8 cultures. Add 10 microliters of freshly thawed luciferin. The luciferin is light-sensitive, so protect the luciferin medium from light by storing the falcon tube in a dark 37 degrees Celsius heating chamber. Next, sterilize all instruments, cover glasses, and other material to be used in the procedure. Spray all non-sterile equipment with 70% ethanol. Expose the instruments and the greased petri dishes to UV in a sterile hood for at least 30 minutes. Another preparatory step is to set up the vibratome and attach a sterile razor blade. The equipment is now set up for the surgical procedure. After anesthetizing and decapitating a mouse, remove the eyes from the head with a pair of scissors. This prevents straining or exciting the optic nerves, which helps avoid damage to the suprachiasmatic nucleus, or SCN. Use a fine pair of scissors to remove the last cervical vertebra and to remove the skin. Make 2 cuts in the skull bone along both sides of the skull, thus making a removable lid. Open the skull with a micro-rongeur tool. Use the rongeur with an upward motion, never pressing down on the brain with the tool in order to prevent damage to the caudal SCN. Remove all bone until the olfactory bulbs can be seen. Use a fine micro-dissecting spring scissors to carefully cut through the optic nerve between the hemispheres and the olfactory bulbs. Place a petri dish filled with 50 to 100 milliliters of cold HBSS under the mouse's head. Turn the head upside down and let the intact brain start to fall out. Cut other cranial nerves as necessary on the opposite side from the optic nerve. When detached, the brain falls into the petri dish, which rapidly cools the brain. Use a spoon to transfer the ice-cold brain to a sterile cutting surface, such as a glass petri dish lid turned upside down. Remove the cerebellum by making a perpendicular cut with sterile razor blades between the hemispheres and the cerebellum. Apply superglue on the dry platform of the vibratome. Insert a sharp curved forceps in the rostral part of the brain and pick up the brain. Holding the brain with the forceps, carefully dry off the HBSS on sterile filter paper. Fix the hemispheres onto the glue on the platform with the rostral tip upwards and ventral surface closest to the cutting blade. Attach the platform in the vibratome holder and immediately fill it up with cold HBSS. If the perpendicular cut is made properly, the hemispheres should stand straight up. The goal is to cut coronal slices containing the midregion of the bilateral SCN. Using the maximum speed of the vibratome, start cutting off 500 to 800 micrometer thick sections of the hemispheres until reaching the commissure. Watch as the optic chiasm gets wider, accompanied by reduction of sections to 100 micrometer thickness. Continue slicing down in the caudal direction, slowing down the horizontal manual movement of the blade until the two SCN nuclei start to appear. Use a magnifying glass if necessary to visualize the nuclei. When the level of the SCN has been reached, cut an SCN section of 250 micrometers. Fill a lid from a medium-sized petri dish with cold HBSS. Using a soft brush, lift and transfer the SCN brain section to the petri dish. Place the petri dish under a dissecting microscope. The next step is to dissect the SCN out of the slice. Use the microscope to verify that the bilateral SCN is clearly visible on at least one side of the section. Use a pair of sterile scalpels to dissect out a square of tissue, about 1.5 millimeters on each side, containing both SCNs. Cut as close to the SCN as possible without removing SCN tissue. A small piece of the optic nerve must be attached to the explant, but no other nuclei should be included. Cut the tissue containing the bilateral SCN in half to obtain two unilateral SCNs. One unilateral SCN can be used as a control. In a sterile hood with the light off, add 1200 microliters of the luciferin medium to a 35 mm diameter petri dish, and then place a culture membrane on top of the liquid surface. Make sure there are no air bubbles under the membrane. The explants are tiny and difficult to pick up. Therefore, use a 1000 microliter pipette with tip to suck the SCN explant into the tip and press it out on the culture membrane. Discard excess HBSS. For luciferase recording, place only one SCN per petri dish. Seal the greased petri dish with a 40 mm diameter cover glass. Add more grease if the seal is not tight. Transfer the petri dishes to a Lumicycle apparatus placed in a 36 to 37 degrees Celsius light-tight heating chamber to begin culturing. Note the culture dishes and photomultiplier tube detector assemblies mounted inside the Lumicycle chamber. Place the culture dishes below the photomultiplier tubes, approximately 1 to 2 centimeters. The photomultiplier tubes detect and amplify the luciferase-induced bioluminescence. The Lumicycle software tool is used for data acquisition. Photon counts are integrated over 1 to 10-minute intervals to get high resolution of the gene protein expression. The circadian expression of the gene protein is analyzed to determine phase, period, and amplitude of the rhythm. This figure shows bioluminescence recordings from healthy and non-healthy SCN tissue cultures. These mice were held in 12 hours light, 12 hours dark prior to the removal of their SCN tissue. The black line shows the circadian rhythm of the bioluminescence recordings of period 2 luciferase expression in a healthy SCN slice. The protein oscillates with a circadian variation in which the maximum expression of the protein occurs at Zeitgeber time 12 to 13, or the onset of darkness. Thus, the phase of the gene rhythm is dependent on the light-dark schedule in which the animal was kept before sacrifice. The blue line shows bioluminescence recordings from an SCN culture that dried out after the culture dish was opened at day 4, indicated by the arrow, and not resealed properly. The red line shows bioluminescence recordings from a non-healthy SCN culture. This figure uses the SCN culture in a pharmacological experiment. The figure shows a trace from a culture that was treated with an HCN ion channel blocker at the time indicated by the first arrow. Over the course of 2 to 3 days, the ion channel blocker reduced and then stopped the circadian oscillation. At the second arrow, the drug was washed out and replaced with conditioned control medium.
While attempting this procedure, it's important to remember to work quickly but carefully in order to keep the SCN tissue viable. It's also important to standardize your vibratome sectioning so that the same level of the SCN is obtained each time.
After watching this video, you should have a good understanding of how to slice and dissect the SCN, culture the SCN organotypically on a membrane, and monitor clock gene or protein expression as luciferase-induced bioluminescence in the cultures.